All right. Hi, everyone. This is Emily Clark at Quasi. I am the training coordinator at Quasi, and I wanted to welcome you all to the kickoff of the 2015 Spring Cyberseminar series. So I'm going to turn it over to our host, Stephen Brantley, at the Joseph W. Jones Ecological Research Center to introduce the series and introduce our speaker today. Stephen?
Thanks, Emily. Welcome to the Cyberseminar series. Our topic, as you can see on the screen, is evapotranspiration, and we're really going to do sort of an overview of the topic at various scales and focus on various fluxes. I am an eco-hydrologist at the Jones Center, so my background is more in the terrestrial sector and plant physiology. But what we're going to hear about today is how some of those upland fluxes.
It was cabin, I think it was 304.
We're going to hear about this from Peter Caldwell. Peter is, let me go to the next slide first. I wanted you just go ahead and give a quick overview of the rest of the session. Next is John Van Stan at Georgia Southern University. And we have Pamela Nager after that, and then Adrian Harpold and Paul Stoy. We'll really cover a pretty wide variety of topics, all the way from eddy covariance to interception, all the way to stream flow. Today's speaker, as I was saying before, is Peter Caldwell. He is a research hydrologist at the Coweeta Hydrologic Lab, and he has been working on this for about 1.5 years now. His background, he went to NC State University.
Great, thank you very much.
Also worked at the Eastern Forest Threat Assessment Center of the Southern Research Station of the U.S. Forest Service before coming to Coweeta, and he has quite a bit of experience in modeling watershed processes. And so today he'll be sharing some insights from the long-term records at Coweeta. So Pete, if you'll take it away.
Sure, thanks, Stephen, and hopefully everybody can hear me okay. I'm going to pull up my presentation. There we go. Hey guys, so again, I'm Peter Caldwell, research hydrologist at the Coweeta Hydrologic Lab. If you don't know anything about Coweeta, I'll by the end of this talk today. So as Stephen mentioned, I've been for about 1.5 years now, and getting really close, I think, to submitting this work. And we wanted to really provide and get your thoughts on it. And really, it's kind of one of the Coweeta is one of the do look at long-term and evapotranspiration and the long-term is that I'm going to share with you today. So that's what I'll be talking about. I'd like to acknowledge my co-authors here. Kelsey Four-Miniot is our project leader here. Lab. He's a research psychologist. Brantley. Elliott is also here at Coweeta. Hydrologist at Coweeta. Plank, who is at the Coweeta lab. So the U.S. Forest Service has a long history of water management. We've had several legislation that have enabled going back to the Organic Act of 1897, which stated that no national forest should be established except to improve and protect the forest within the boundaries for the purpose of securing favorable water flow. We've had several pieces of legislation like 1897, but in each of these, we've always had a component that dealt with flows and protecting water quantity and quality. So in response to this charge, the Forest Service has established 80 experimental forests and ranges across the United States, which you can see here in this map. And the purpose of these experimental forests were to provide places for long-term science and management studies in the major vegetation types across the U.S. And so the Coweeta Hydrologic Lab is located in the Southern Appalachian Mountain region in North Carolina. So again, the Coweeta Hydrologic Lab has been around a long time, and you know, always central to our mission has been conducting basic research on how forests and water interact. And I love this old sign here that was posted at Coweeta back in the 50s, and it kind of gets at that mission. So coming up with practical methods of forest land management to maximize water production consistent with region-wide interest in flood control and industrial, municipal, and recreational use of water. So it's really interesting to hear, you know, what were the issues of the time, but, you know, certainly then, as is now, water has been a central part of the mission of the lab.
Hi, Pete. Sorry to interrupt you. Could you just back up a couple of slides? It looks like there was a problem with the audio, and
Sure.
Folks missed it. Could you just do a quick recap of those first couple of slides?
Certainly. So basically, I was going into some of the history of the U.S. Forest Service and our enabling legislation, going all the way back into the early 1900s, and basically, you know, throughout our history, we've always had a mission of protecting water quantity and quality. Can you all hear me better now? Okay, I'm seeing some yeses. Okay, so in response to this, we've got 80 experimental forests and ranges across the United States. And the Coweeta Hydrologic Lab is one of these. We're located in the Southern Appalachian Mountain region in North Carolina. And the mission of the Hydrologic Lab at Coweeta has always had a central focus on water, and this old sign that was posted at Coweeta really captures some of the issues of the time, and some of these are still relevant today. So the Coweeta Basin in the lab was established in 1934. The basin is a little over 1,600 hectares. And what you see here in this map, the yellow lines depict the perimeter of the basin, but also several smaller first and second order catchments that the basin has been delineated into. And some of these basins have been treated over the years in a variety of forest management experiments, and others have been used as controls and have not been manipulated since the establishment of the lab. So we currently have 16 active weirs measuring stream flow at 16 of our subcatchments within the basin. Again, some of these going way, way back into the mid-1930s. We also have 5 climate stations distributed across the basin where we're measuring air temperature, relative humidity, wind speed, direction, and that type of thing. The climate station that you see down at the bottom there near the outlet of the basin is Climate Station 1, and this climate station has been there since 1936. So measuring continuous climate information since that time. We also have 10 rain gauges distributed across the basin measuring precipitation, both with standard rain gauges and recording rain gauges. In addition to the hydroclimatological data that we're collecting, there are 676 vegetative plots organized in transects across the basin. In these plots, we've put numbers to species composition and forest structure metrics. About 280 of these plots have been measured 4 times since 1934, roughly about every 20 to 30 years. So a long-term record of how the forest community has been changing at the lab. So some of the classical water yield and evapotranspiration studies have been conducted at Coweeta include management experiments where we might clear-cut a watershed and look at the impact on water quantity and quality. So at the top of this slide, you can see watershed 7. This watershed was clear-cut and cable-logged back in the mid-1970s. And the chart you see there on the right is where we've shown through the paired watershed approach, or the before-after controlled watershed approach, how water yield has changed in response to that clear cut. So we see increases in water yield due to decreases in evapotranspiration immediately after the harvest, but within 4 to 5 years, the effect of this this harvest essentially goes away in terms of annual water yield. Below that, you see one of our other classical experiments where we've converted from the native deciduous forest to white pine, a coniferous species, and this took place in watersheds 1 and 17. And you can see in the charts on the right there that water yield increased after the clear cut of the deciduous forest, but over time, because the coniferous trees are transpiring year-round and intercepting rainfall year-round, we're seeing less water yield in those watersheds than we would have expected under the deciduous forest conditions. So why is this important? Well, the Southern Appalachian region are headwaters for drinking water supply for much of the southeast. So we just completed a study last year where we looked at how forests in the south are contributing to water supply for a variety of drinking water intakes across the region. And so the Southern Appalachians are 81% forested, and they're providing about 23 billion cubic meters of water per year to drinking water intakes in the region. There's about 588 intakes serving 336 communities. About 10.7 million people are getting some of their water from the Southern Appalachians. About half of these are getting more than 20% of their water from the Southern Appalachians. So the region is really important for drinking water supply, and so certainly examining long-term trends in water yield and evapotranspiration are important for planning purposes for the region, especially in the face of climate change. So some of the things that we've learned at Coweeta by examining our long-term data are that, number one, we're seeing increasing air temperatures and increasing precipitation variability. So these are measured at that climate station I mentioned at the base of the basin, and you can see that the air temperature on the left there, that air temperature has been increasing since about 1980. And precipitation, while the mean hasn't really changed, we're seeing much more variability in our precipitation. So we're seeing more frequent wet years and more frequent dry years. So the question is how does this affect water yield and evapotranspiration? In addition to changes in climate, we've had several major disturbances in the forest that have occurred over time. Some of these are related to climate change, for example, droughts and hurricanes and major flood events. But others are related to forest invasive species and pests, for example, the chestnut blight that wiped out the American chestnut from the region in the 1920s and 30s, as well as the hemlock woolly adelgid that decimated the hemlocks more recently from beginning in about 2003. So lots going on in the forest, even though much of our watershed is unmanaged. There have been some natural and I guess unnatural disturbances that have occurred that could be affecting ET. So in response to these disturbances, we have noted some changes in the forest species composition. So using the data from those vegetative plots that I mentioned, you can see the crash of the American chestnut at Coweeta. These are the black dots that you see here. In the 1930s, this is the most important species in the basin, and that decline ever since then, since the chestnut blight. You can also see this with the eastern hemlock. These are the brown squares, kind of near the bottom, and you can see the decline in hemlock in the more recent sampling period. There have been some winners in this story. Red maple, the green squares on the rise, as well as tulip poplar, which are the pink circles in this chart. So our forest is changing species as a result of these disturbances. And although Coweeta is an experimental forest, these disturbances are representative of what's going on across the Southern Appalachian mountain region. So the questions we've been wanting to answer here are, have there been any changes in water yield in some of our unmanaged forests in the Southern Appalachians? And we want to identify what could be causing those changes in water yield. Is it related to precipitation or evapotranspiration, or both? And if it is related to evapotranspiration, how much of this is related to changes in energy and atmospheric conditions, in other words, potential evapotranspiration, or how much of it could be related to these changes in the forest species composition and structure? So as I mentioned before, several of our classical experiments that have gone on at Coweeta have been using the paired watershed approach. This is where we have two watersheds, usually side by side, and we will treat one of the watersheds, for example, a clear cut, and we compare the change in water yield in that watershed to its neighboring control watershed to determine what were the impacts of this treatment. And so we have 6 control watersheds at Coweeta, and you can see those here on the map. Those that you see on the left are at a higher elevation than those on the right. And so they generally have higher precipitation, as much as 20% higher precipitation at the higher elevations than at the low elevations. And so we see much higher water yield and also much lower evapotranspiration at the high elevations due to decreases in air temperature. So the way we went about quantifying changes in annual water yield was to use the AutoRegressive integrated moving average time series modeling. This is also known as ARIMA. This is a statistical method that's a type of regression model that you can use to fit time series data. And with this type of modeling, you can account for autocorrelation or memory in your data, so how well is one data point correlated to data points before that data point. And you can also account for external perturbations that may be affecting the time series, and we call these interventions. So for example, you could have a step intervention when you convert a forest to an agricultural field that could be changing the relationship between precipitation and water yield. Or you could have more gradual interventions like a ramp function that could occur as a forest ages over time and species change over a longer period of time. And so when you apply this methodology, you consider a range of plausible models and you select the best model, considering how well it fits the data and how simple the model is. And the Akeiki's information criterion is a way of quantifying both goodness of fit and parsimony of the model. So we considered 4 different models, and I'm showing examples here for Watershed 2. The first model in the top left was just predicting water yield using the mean over the entire time series. The second model we consider, which is the top right, includes the mean with a ramp intervention or a gradual perturbation at the start of the time series. The third model is in the bottom left. This is a mean with a ramp intervention at the end of the time series. And then finally, in the bottom right is a model that includes the mean with ramp interventions at both the start and the end of the time series. And so when we ran these models, we considered a range of years to determine the breakpoints in these interventions. And then we compared each of the 4 models against each other using the Akeiki's information criterion. And when you would do this for Watershed 2 here, you would choose the model that has the lowest AIC value. And so for watershed 2, this is the model that has the intervention at the beginning of the time series. And so this is the best fitting, most parsimonious model to predict water yield in this watershed. So using this approach across all 6 of the control watersheds at Coweeta, we discovered that we have seen significant changes in water yield in our low elevation watersheds, which you can see here across the top of the chart. So again, watershed 2, the best fitting model has a single intervention at the beginning of the time series. Watersheds 14 and 18, which are north-facing watersheds, have increasing water yield at the beginning of the time series and then decreasing after the mid-1970s or so. The high elevation watersheds, we were not able to detect a significant change in water yield. So the model with just the mean was the best fitting, most parsimonious model for these watersheds. So looking again just at the interventions and the model predictions for water yield here, you can see that in watershed 2, the increase in water yield occurred from the beginning of the time series in 1938 up until 1949. Watersheds 14 and 18, there was a break point about 1974. And the changes in water yield, all of the changes I'm showing here are significantly different from 0, so p-values less than 0.05. And these water yields these changes in water yield are fairly significant. So looking at watershed 14 from 1938 to 1974, there's an increase in water yield of 73 millimeters per decade, which is about 30% increase from 1938 to 74. And after that time, water yield decreasing 64 millimeters per decade, or about 22% between 74 and 2013. So we do think that there have been some changes in the long-term water yield in the control watersheds at Coweeta. And so now we wanted to look at what could be causing these changes. How much of this could be related to changes in precipitation? How much could be related to changes in evapotranspiration? Using the same approach with the ARIMA models, we found that changes in precipitation were not significant. The best fitting, most parsimonious model for precipitation included a single ramp intervention at the beginning of the time series. But you can see that the p-value on this is greater than 0.05, so this change is not significant. But certainly you can see the increase in variability in precip at this rain gauge, beginning somewhere around 1980. Much more variability, more wet years, more dry years than beforehand. Also applying the ARIMA methodology to evapotranspiration, this is estimated by taking the difference of precipitation and water yield in each of the watersheds. When we do this, we see that there have been significant changes in evapotranspiration across all of our watersheds, low elevation and high elevation watersheds. In general, decreasing evapotranspiration at the beginning of the time series, some time of relatively stable evapotranspiration, and then increasing again. So looking at the magnitude of these changes in evapotranspiration, looking at watershed 14, for example, evapotranspiration decreasing from 1938 to 1974 by about 31 millimeters per decade. And then after 1980, increasing by about 37 millimeters per decade. And this pattern is fairly consistent among all of the watersheds, with the exception of watershed 27, which has only the single intervention at the beginning of the time series. So yes, I think we can say that there have been some changes in precipitation, at least in terms of variability, as well as evapotranspiration that we're able to estimate by subtracting runoff from precipitation. So now we wanted to look at what could be causing these changes in evapotranspiration? Is it related to changes in the energy and atmospheric conditions or potential evapotranspiration? Or could it be related to the forest species, composition, and structure? So at Climate Station 1, we have an evaporation pan that we've been measuring pan evaporation daily since 1936. And so using this evaporation data as a proxy for potential evapotranspiration, we see that we have had some significant changes in potential evapotranspiration over the period of record. From 1938 to 1949, potential evapotranspiration decreased by 50 millimeters per decade. And beginning in 1997 and going until the present day, or 2013, potential evapotranspiration increasing 46 millimeters per decade. So certainly there is some evidence here that suggests that our PET has been changing over time, and this pattern of potential evapotranspiration kind of mimics what we see in the P minus Q estimate of evapotranspiration. So we wanted to be able to model the effects of climate on water yield. And so we assumed a model where we could calculate water yield as a function of annual precipitation and annual potential evapotranspiration measured with the evaporation pan. And so you can see our model hypothesis here. Coefficients are calculated using the ARIMA procedure. The A sub t term in this equation is the error. You can see the results of this modeling approach across all 6 of the watersheds at Coweeta. First, you can see that the model does a pretty good job of explaining the variability in the measured water yield with R-square values of 0.88 to 0.94. I'm seeing some conversation about the sound quality. Is there something I can do differently?
I don't think so. I think you sound good to me.
Okay.
Mm-hmm.
Okay. All right. Great. Thank you.
Yep.
So something else that we found interesting in looking at the model parameters here is that these coefficients, they kind of make sense, at least for all watersheds except watershed 36, one of our high elevation watersheds. So the coefficient on precipitation in the 85% range, this is roughly equivalent to what we know is the throughfall in our forest here at Coweeta that we've measured over the years. So that kind of makes sense and passes the gut check. The coefficient on potential evapotranspiration also kind of makes sense. So there's a lot of things rolled up into this coefficient. One is that we have different climate regimes in our watersheds compared to where our climate station is measuring pan evaporation. We also have topography rolled into this, watershed aspect and slope, as well as the difference between vegetation, evapotranspiration from vegetation, and evaporation from the evaporation pan. But when you look at these coefficients relative to each other across the watersheds, you can see some patterns that make sense to us. So for example, watershed 2 is a south-facing watershed, has a higher coefficient on potential evapotranspiration than does watershed 14, which is a north-facing watershed. And you can also see as you go up in elevation, this coefficient on potential evapotranspiration decreases with elevation. So this model kind of makes sense, at least conceptually. And just as an example of how the model fit looks, this chart is showing observed and predicted water yield for watershed 14. Predicted water yield in the lines and the circles are the observations. And you can see that overall the model is predicting the observations fairly well. But you can also see some patterns in the error here. For example, near the middle of the time series, this model is under-predicting water yield compared to the observations. And so when you look across all of the watersheds at Coweeta and look at the residuals of this model, you can see that the residuals have a nonlinear association with water yield, so they're not constant over time, which is something that you would want. So what these are telling us is that the model is under-predicting water yield at the beginning of the time series, kind of over-predicting water yield near the middle of the time series, and then under-predicting again at the end of the time series. So we wanted to try using the same model, but then adding interventions to the model, similar to what I showed you before. And so when we run this model across all the watershed with interventions, we get interventions that look like the chart you see there in the bottom left across all of the watersheds. In all cases, these interventions improve the model fit. They're all significantly different from 0. And what they represent is a change in water yield after you're accounting for changes in precipitation and potential evapotranspiration. And they represent a change in water yield of about 5 to 10%. So this is a small number, but still significant. So this slide is showing how these interventions affect the predictions of ET. So I'm showing here a 5-year moving average of precipitation minus water yield, or ET. The gray line are the observations. The dotted lines are the predictions without the interventions. The black lines are the predictions with interventions included. So starting with the dotted lines, the predictions without interventions, you can see that the model underestimates evapotranspiration at the beginning of the time series, overpredicts during the middle of the time series, and then somewhat underpredicts towards the end. When we add the interventions to the model, however, we split the difference on the beginning of the time series and have a much better fit than we had without the interventions. And then after our breakpoint in 1974, the interventions help us to fit the data much better than without the interventions. So what could these interventions represent? Well, one thing that could be going on is that we could be changing how we're measuring water yield and precipitation over time. And this is quite possible. We have gone digital at Coweeta. We've gone away from paper charts, but overall, our methods haven't changed significantly. We're using the same weirs and same rain gauges and the same locations. And even if this was the cause of all of this, we would maybe expect a step change in water yield in ET as opposed to these gradual ramp changes. Another thing that could be causing this is changes in climate that we're not accounting for with an annual time step model. So for example, changes in the seasonal timing of precipitation and the growing season length and that type of thing. We looked at this with a monthly water balance model, which accounts for changes in the seasonal timing of precipitation, and we also see that modeling approach has a nonlinear relationship between the residuals and water year. So we don't think that that's it. Another thing it could be is some vegetation response to a non-climate driver, for example, ozone or carbon dioxide concentrations in the atmosphere that we're not accounting for in the model. And we've not explored this fully, but we don't know of any non-climate drivers that follow this increasing, decreasing pattern of water yield and evapotranspiration. So what we're proposing is causing these changes in evapotranspiration explained by the interventions is changes in forest species composition and structure. We know that the forest is getting older, and Stephen has done some work at Coweeta and other places in the region relating canopy interception with forest stand age. And you can see that interception as the percent of gross precipitation increases as the stand gets older. So potentially increases in canopy interception could be explaining the increases in ET over time, at least since the 1970s. We also know that water use varies by species. There's been a lot of work done at Coweeta by Chelsea Miniot and others using the sap flux method to measure actual plant transpiration among trees. And this chart shows what the effect of species on plant water use. Trees with a diffuse porous and tracheid xylem anatomy, for example, red maples and tulip poplars in the case of diffuse porous, and white pine and hemlock in the case of a tracheid, have much larger functional sapwood area, which you can see by the dye and the cross-section there of the stem, and therefore they use more water for a given diameter than do other species with a ring porous xylem, for example, red oaks and chestnut oaks, which are shown in red on this graph. So for a given diameter, oak trees are using less water than diffuse porous species like red maples and tulip poplars. Using our vegetative plot data over time, we are seeing that we're shifting to some of those heavier water users, so species with the diffuse porous xylem. So this chart is showing the mean basal area among permanent plots in watershed 14, and you can see that the basal area of the diffuse porous species has been increasing steadily over time. Ring porous species took a hit between 1934 and 1970. This is related to the chestnut blight. Increasing diffuse porous species in the watershed. Something else you can see here on the far right is the evergreen shrubs. These are rhododendron and mountain laurel. These are also diffuse porous species, but they also occur in the subcanopy of the forest. These evergreen shrubs are on the rise as well, and so they can increase interception by the canopy by existing in the subcanopy, and they're also diffuse porous species and use a good amount of water for a given size. So we are seeing some changes in the species composition that could be explaining these changes in evapotranspiration. And so what we're seeing is that some of these forest metrics that we can calculate or glean from these permanent plots reflect the interventions and changes in evapotranspiration. So here I'm plotting watershed 14 P minus Q ET. The gray line are the observations. The black line is the 5-year moving average. I'm also plotting metrics of forest species or forest structure with the symbols in this chart. All of these calculated relative to 1934, and so you can see that, for example, leaf area index is following a similar pattern to what we're seeing in the evapotranspiration observations, similarly with foliar biomass and total biomass. So potentially these changes in forest species could be explaining what we're seeing in evapotranspiration. So in summary, we have seen changes in water yield at unmanaged forests in the Southern Appalachians at Coweeta. We see that water yield has increased 30 to 55% in our lower elevation watersheds until the early 70s, and then decreased by about 22% from the early 70s to 2013. There have been some changes in precipitation and potential evapotranspiration that explain some of these changes. But we also think that changes in forest species composition and structure also likely play a role on water yield and ET. So some ongoing work that we have going on at Coweeta, we have a USDA AFRI grant where we're looking at the impact of forest stand age and structure on forest water yield. So the data I've been showing you have been a single watershed over time. In this case, we're going to be taking the space for time substitution approach, and we have 5 sites across the region of different forest stand ages, and we're quantifying the component water fluxes of the water balance in each of these stand ages. What we'd like to do is take this theoretical graph you see in the bottom right and fill that in with actual data to understand how forest water use varies over time and changes over time. So with that, I'd like to thank you for your attention. Thank you to our funding sources, the USDA Forest Service Southern Research Station, the National Science Foundation LTER program. Coweeta is an LTER site through the University of Georgia, as well as the USDA AFRI foundational program. And thanks especially to all of the scientists and technicians who've contributed to this long-term data set and made this type of analysis possible. So with that, I guess we can take questions.
Yeah, so we will turn it over to the participants, and if you have a question, please type it into the chat box, and Stephen and I will relay it back to Peter.
Okay. Yeah, we have several people typing now, and I know Pete can see these, but I'm just going to kind of repeat it for everyone's clarification. Sharon Lopez asked, the higher and lower ET periods correspond with negative and positive phases of the PDO. Have you explored teleconnections?
I have not done that personally. I know that a previous hydrologist here at Coweeta, Mark Riedel, looked at some of this, and certainly that could be true. So the changes in ET, or at least potential ET with evaporation pan, we would expect to see this effect in our potential evapotranspiration, and so that could be related to the PDO. Okay, and we can follow up later if that is not satisfactory. But Nicholas Zegra, and I think I know the answer to this already, but have hydrology data at the outlet of the Coweeta Basin been analyzed for changes? What about at larger scales, i.e. the Little Tennessee watershed? Yeah, that's a good question, and that's actually kind of how I started this work, was looking at it at the larger scale at the Little Tennessee. And the Little Tennessee River, the data go back to 1944, and we can see some of these effects going on at that scale. I'm struggling a little bit right now with determining sources of climate data to use at that scale, but we certainly think that at least some of what we're seeing here is also occurring at larger scales, particularly at the end of the time series. I've looked at data going back to 1960 and forward, and we see these increases in evapotranspiration since that time, since about 1980 in the Little Tennessee. Okay. The next question, do we have anything published that we can point to for any of these results? Not yet. I'm glad to hear that there's interest. I'm in the process right now of writing this up, and we hope to get this paper submitted within the next month or so. So we can make this publication known once we get to that point. Okay. Steve Burgess asks, do you have any pit rain gauges in the basin? I don't believe we do. I guess I'm not sure what is meant by a pit rain gauge. Each of our rain gauges are established in a clearing in the forest, and they're all about a meter or so off of the ground. Yeah, I'm not either familiar with that type of rain gauge either. Maybe some clarification later, but Chuck Slaughter has a question on the earlier slides. Watershed 14 and 18 are controls, and they are, let's see, slides of Q and ET indicate both water yield and ET are now approximately back to 1940 values. So I guess, can we explain that? Yes, yes. So watershed 14 and 18 are controls, and I believe stream flow in watershed 2 began in kind of in the middle of 1936. Watershed 14 in the middle of 1937, and all this work was done on a water year that began in May, And so basically we started those time series in 1938. So we didn't use all of the data, but in order to use that May to April water year, we started about that time.
Okay, and next we have a comment. You all should study WMO standards.
I guess we should.
Okay, so there's a little more typing happening, so we'll see if anyone else has a question.
Okay, someone is asking about the paper referring to quantified water production for the Southeast. Pete, do you want to give the kind of rough citation for that?
Yes, this is a U.S. Forest Service General Technical Report, or a GTR, and the Forest Service has a website called Tree Search where you can search for Forest Service publications, and if you search for me as the author, Peter Caldwell, you can see all the publications that I have in there, and that publication was published in 2014. It was called something like Quantifying the effect, or quantifying the contribution of forested lands to water supply in the Southeast.
Okay, and we can reference Steve Burgess's comment about the Seek et al. Water Resources Research paper. I'm going to move on to the next question. Let's see. I think there's a little bit of more interest in the fact that the 2014 values of Q are about the same as the 1940s. Can we elaborate on that a little more? About, I guess maybe the question would be, why would there have been a decrease in Q and then or an increase in Q and then a decrease over time? What was the explanation for that increase in Q?
Okay, well, the increase in Q after the 1970s, part of that was probably related to, or actually the decrease in Q, increase in ET after the 1970s. Part of that is related to the increase in potential evapotranspiration that we were able to observe from our pan evaporation data. But also part of it, we think, is related to the changing forest species composition and structure, so the increase in diffuse porous species in the forest, as well as just the general increase in age of the forest over time, an increase in interception.
And as Justin points out, there may have been some effect early on of chestnut loss in that in some of those earlier data. Would that be correct?
Yeah, I think that's. I think we could probably conclude that the loss of the chestnut in the beginning of the time series led to a decrease in ET and an increase in water yield, and then over time, the forest is recovered, and we've got more of the diffuse porous species like red maple and tulip poplar dominating the species mix and increasing ET and decreasing water yield.
Okay, we have another question from Mohammed. Based on your experience, do you have any recommendation in terms of data collection and instrumentation for a newly established experimental watershed? And the example is Kings River Experimental Watershed in California.
Well, there could be a lot rolled into that question, and we could certainly talk to Mohammed offline and talk about our experience here at Coweeta.
Yeah, and I think a lot of it might depend on the size of the watershed and sort of the land use questions that you would want to get at in terms of what sensors you would need.
Certainly.
Steve, thanks you for an excellent presentation and suggest investigating Hearst, Sameka, and Black, 1965.
Okay.
And then we had another comment, but it was deleted. Let's see. Okay, this is an excellent question. Have you observed peak flow changes? And I don't think you've gotten into this at all, have you?
I have not. I was going to defer this to you, Stephen.
Well, I would only comment on the experimental watersheds, the reference watersheds, I should say, that have experienced loss of hemlock woolly adelgid. We did see some very, very slight changes in peak storm flow for just a couple of years, but we haven't really undertaken this kind of extensive analysis of peak flows for the reference watersheds other than in relation to hemlock. That might be it.
Okay, great. Well, if those are the questions that we have, we can go ahead and end here. So, Peter, thank you so much for presenting today.
Thank you.
And Stephen, thank you for hosting. And again, I'd like to remind everyone that next week we're going to have John Van Stan from Georgia Southern University. So April 3rd at 3 p.m. Eastern Time will be the next presentation in this series. So thank you all, and have a great weekend.
Great. Thank you all for joining us. I'll reiterate that. And thank you, Pete, very much for volunteering.
Thank you. Thanks for the opportunity.
Sure.